the origin is from RNA transcribed from distinct gene. Okay, I'll show you how microRNA, the biogenesis of microRNA later. Okay, so small interfering RNAs, in terms of the cleavage, it will cleave RNA duplex or single-stranded RNA that forms long hairpins, while microRNA cleave single-stranded RNA that form short hairpins. Okay, so this is the difference in terms of cleavage. Okay, in terms of action, okay, a small interference RNA trigger degradation of mRNA, while others inhibit transcription. Okay, while microRNA also trigger degradation of mRNA, but inhibit translation instead of transcription. Okay, so the target, which is the expression of the genes that they control, is actually for sRNA is the genes which they will transcribe, but microRNA regulates the target genes are others from which they were transcribed. Okay, so these are the differences between small interfering RNA and microRNA. Okay, I'll show you the diagram. Okay, so microRNA. Okay, so the origin, you have DNA, okay, which is double-stranded. For small interfering RNA, you have small double-stranded RNA. Okay, and through transcription for microRNA, they akan form an inverted repeat in the DNA. Transcription through an inverted repeat in the DNA to produce RNA. Okay, so then this RNA, okay, akan fold to produce what we call double-stranded RNA. Okay, they form loop. Okay, so the next step, the double-stranded RNA is cleaved by the enzyme, what we call as dicer. Okay, so the dicer will cleave this double-stranded RNA yang form through the formation of loop tadi lah. Okay, so one strand of the microRNA will combine with protein. Okay, it will combine with protein, okay, to produce what we call as RISC ataupun RNA-induced silencing complex. Okay, so the RISC akan pergi ke target that gene or mRNA yang dia nak regulate tu lah. Okay, dia akan base pair, imperfect base pairing of the mRNA, okay, and inhibits the translation, okay, of the mRNA ni lah. This is the target gene yang dia nak regulate lah. Okay, so this is the mechanism of microRNA. Okay, while small interfering RNA from double-stranded RNA, okay, which may arise from the RNA viruses or long hairpin. Okay, so then the double-stranded RNA, okay, is cleaved by the enzyme dicer, sama. Okay, and then the protein will combine with the yang tadi tu, with the protein to form protein will form with the siRNA yang produce tadi tu, okay, to form RISC, okay, and dia akan this RISC complex akan okay, so it different from microRNA which is imperfect base pairing. You akan nampak bukan semua sequence akan base pair dengan mRNA, but for small interfering RNA is a perfect base pairing between the mRNA and the RISC. Okay. Alright, so then it will degrade the mRNA. Okay, right. So so this is the difference between siRNA and also microRNA. Okay, so I will focus only giving example on microRNA. Okay, so what are microRNAs? Okay, they are actually 21 to 24 nucleotide. Okay, dia ada macam ATGC, for example, 21 to 24. Riboregulators, actually this microRNA was first discovered, okay, in nematode. Okay, so it being discovered in this nematode, C. elegans. Okay, so microRNA are processed from imperfectly packed hairpin precursor, okay, which are generally produced from transcriptional unit that are distinct from those of protein coding genes. Okay, and mature microRNA target protein coding mRNA. Okay, so they target this protein coding mRNA to regulate the expression of this mRNA of the genes. Okay, so in animals, microRNA often display limited complementarity to multiple sites in the 30 untranslated region of the target message. But in plants, okay, the microRNA generally display near perfect complementarity, okay, to a single site within the target message. Okay, so and can direct the cleavage at this site. Okay, so this is the difference between microRNA in animals and also in plants. Okay, so this is the biogenesis and function of microRNA. Okay, so you start daripada atas. So to produce microRNA, the microRNA will be transcribed. Okay, will give rise to transcript. Okay, that contain an imperfectly paired hairpin region. Okay, so this is a hairpin region lah. Okay, and then this transcript, okay, you have microRNA gene being transcribed. Okay, and this transcript are processed by DCL1 ribonuclease and perhaps assisted by the HAN1 and HYL1, okay, proteins, okay, into an imperfectly paired double-stranded RNA of about 21 nucleotide that has 2 nucleotide 3' overhang. Okay, so you nampak DCL1 through transcript processing with the help of HAN1 and also HYL1 proteins, okay, akan form double-stranded RNA, okay, nampak double-stranded RNA, tapi ada overhang, okay, dalam 2 nucleotide, okay, alright, yang overhang, okay, dia tak perfectly ni lah pair, okay. So, and then the next step, and identify helicase aids in the selection of the single strand. Okay, so dia pilih satu daripada 2 ni, dia pilih satu single strand of RNA that is incorporated into the RIS. Okay, right? Alright, and then within the RIS, the microRNA serves as the specificity determinant to reduce target protein level. Okay, either by cleavage of the target mRNA. So this is the target gene of the mRNA of the target gene. Okay, dia menyebabkan this RNA bring cleavage. Okay, bring cleave, ataupun it can reduce the translation of the uncleaved target mRNA. Okay, so by either by cleavage of the target mRNA or by the reduced translation, okay, it will result in reduced target protein levels. Okay, so this is how microRNA regulates gene expression. Okay, any question? Ada soalan? Clear ke?
No, Doctor.
Yeah?
Yeah.
Alright, okay.
Ada.
Alright, sorry. Saya tahu susah sikit kan plan genetics ni. Okay. Alright, so a little bit of explanation lah, which I will give you examples lah. Okay, right. So as I mentioned, kita akan bagi example in Arabidopsis. Okay, because we already explained the importance of Arabidopsis. Okay, as model organism for plant studies lah. Okay, alright. So the information that we have about microRNA implants are based on the studies that scientists done on Arabidopsis. Okay, so the dicer enzymes tadi tu, yang ni, okay, DCL1 ni semua dicer enzymes are characterized, okay, the characteristic of dicer enzymes, they have domains. Okay, so they have domain of amino terminal dex H box RNA helicase domain. Okay, they have pas domain, okay, which is also found in the argonaute proteins. Okay, they have the dicer enzyme also have 2 RNAs, 3 domains. and they also have carboxyl terminal double-stranded RNA binding domains. Okay, you don't have to remember this. It's just explanation lah. Okay, so in Arabidopsis, it has 4 dicer-like proteins, DCL3, okay, which is needed for endogenous sRNA accumulation. Okay, and DCL2 for efficient accumulation of certain virus-derived small RNA, and DCL1, okay, which is needed for microRNA accumulation. Okay, so between plants and animals, okay, there are differences. Okay, so most animal dices lack the nuclear localization signal that is found in Arabidopsis, DCL1. So in animals, what happened? The microRNA transcripts are processed in the nucleus, okay, by the ribonuclease brochure, okay, into the pre-microRNA hairpin, and then it will be exported to the cytoplasm, okay, for dicer processing. Okay, while in plant, in Arabidopsis, the DCL1, which is the closest homologue to animal dicer, is nuclear. Okay, the origin from nuclear. Okay, and processes in the microRNA 159 precursor in the nucleus, suggesting that plant microRNA are actually generated in the nucleus. Okay, which is totally different lah from animals. Okay. Alright, so in addition to DCL1, tadi kita ada HAN1 protein and also HYL1 protein. Okay, so these proteins are required for microRNA accumulation. Okay, and also required for normal development in Arabidopsis. Okay, so in plants, there are possibility that microRNA regulate or modulate hormone signaling. Okay, so this is the function of microRNA. Okay, they also possibility that they modulate hormone signaling in plants. Okay, so whether HAN1 nor H1 are absolutely required for microRNA bioinesis, the double mutant is infertile. Okay, so scientists create mutant of mutant pada HAN1 and HYL1. What they found that the double mutant is infertile. Okay, so they suggest that because mutant kan of these two genes are being mutated, okay, they become infertile. So they suggest that these two genes actually act synergistically together. Okay, so they need each other lah. Okay, so this is the function of HAN1 and HYL1. Okay. And then members of the argonate family also are implicated in microRNA and sRNA functioning. Okay, so argonate proteins are defined by the presence of the PAS and PIWI domains, where the PAS domain is shared with dicer and function to bind RNA. Okay, as argonate proteins reside in the wrist, okay, the diversity of argonate proteins may contribute functional specificity to various wrist-like complexes. Okay, and in Arabidopsis, there are 10 argonate-like genes. Okay, all right. So I will show you video first on RNAi.
Be bold to dream, because our dreams are too good to hold back.
Hello and welcome to today's talk about RNAi
Jadi saya
or
dah tunjuk ke?
RNA interference.
Saya dah tunjuk yang ni kan? Betul ke? Betul ke saya dah tunjuk yang video ni? Ke you terlupa?
Tak ingat.
Okey, saya tunjuk yang lain lah. Ada lagi yang lain kejap.
I'd like to introduce with explaining the
Okay, this is a better video. Okay.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future, perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
Okay, all right. The microRNA ini, RNA ini, okay, it's actually like saya cakap, was being discovered in C. elegans, okay, by the scientist actually Craig Mello. Okay, so dia yang discovered. So they come up with this video, advised by Craig Mello. Okay, alright, alright. So kita continue balik dengan slide. Alright, so these slides, okay, the next slide will give you examples of target of implant, the microRNA target what genes. Okay, so alright, so for example, ni lah macam dia nak validate lah the targets of plant microRNA. Okay, so when a microRNA targets multiple mRNAs, that the mRNAs that they want to regulate, the targeted genes are often members of a gene family lah. Okay, dia katakanlah the target group of gene family. Okay, so and microRNA that are conserved between Arabidopsis and rice also tend to have conserved target. Okay, so if you compare the microRNA between Arabidopsis and microRNA in rice, okay, if these microRNA are conserved between these two plants, okay, they also have to have the tendency to have a conserved target juga. Target is similar. Okay, so intriguingly, plant microRNA display a striking propensity to target the mRNA of transcription factors, okay, that already being established or related to key developmental regulators. Okay, so other targets of microRNA include the dicer and argonaut family, okay, as well as adenylyl L-methionine dependent methyl transferases, F-box proteins, okay, that mediate ubiquitin dependent protein degradation, and also the target of RNA include proteins that are involved in sulfate assimilation. Okay, so these are the example, okay, of different type of microRNA in Arabidopsis, and these are the target protein. Okay, the target protein meaning the protein that they control, regulate. Okay, so for example, microRNA 156 or /157, target proteins are the proteins of squamosal promoter binding. Okay, so ada different type of protein lah. Okay, so you don't have to remember. Okay, so one example is actually where microRNA regulates the development in plant. Okay, so for example, microRNA 165/166 functions to Arabidopsis class 3 homodiomine leucine zipper transcription factor. Okay, also function to regulate Revoluta, Fabulosa, and Fabulosa mRNA. Okay, so this mRNA give rise to abaxial tissues. So they regulate this mRNA that involve in the development of abaxial tissues. Okay, so during leaf development, okay, RLD1 protein, okay, and microRNA 166 are expressed, okay, in reciprocal domains, where this is consistent with the role of microRNA 166 to restrict the presence of RLD1 transcript, okay, within the abaxial tissue. Okay, so they target RLD1 lah transcript. Okay, so this microRNA is expressed in expanding pattern, okay, during leaf development and accumulates in the phloem, which suggests that it is a mobile signal. Okay, and one of the characteristic features of microRNA is that they are evolutionarily conserved. Okay. All right. Intriguingly, okay, this microRNA 165166 actually to be found in class 2 HG transcription factor, not only in flowering plant, for example, in Arabidopsis, rice and maize, but they are also being found in gymnosperms, ferns, lycopods, and also bryophytes. Okay, so this is one example where microRNA regulate development, giving example to leaf development involving abaxial tissues formation. All right, so that is example. Okay, and this example, okay, tadi tu microRNA regulates development. Okay, but at the same time, microRNA also regulate their own biogenesis or their own function. Okay, right. So interestingly, two microRNAs target the mRNA of gene that are implicated in microRNA biogenesis in DHCL1 or in the function Argonaute1. Okay, so for DCL1 mRNA, okay, it is being targeted by microRNA 162. Okay, all right. So unlike many validated microRNA mRNA pair, okay, so you know microRNA akan target mRNA, so the pairing. Okay, so for DCL1, one nucleotide bulge in the DCL1 mRNA is present. Okay, when microRNA 162 pairs with DCL1. Maknanya ada bulge, maknanya tak base pair, complete base pair. Okay, so despite this bulge, which occurs across from position 7 to 8 of the microRNA, all right, the DCL1 mRNA is cleaved in the middle of the microRNA, complementarity site. Okay, and in DCL17 and HEN1-1 plants, microRNA6 levels are reduced, whereas DCL1 mRNA accumulates to a higher level than in wild-type plants. Okay, so this suggests that microRNA 162 is a part of negative feedback loop that control DCL1 levels. Okay, for Argonaute1, okay, the regulation is the expression is being regulated by microRNA 168. Okay. Alright, so these are the example lah. Okay, you boleh baca sendiri. Okay, so I give you two example where one example is involvement of microRNA in regulating development of leaf tissue. And another example is microRNA is being used to regulate their own biogenesis and also their function. Okay. All right. And then we have ribose switches. Okay, another how involved in regulation of expression in plants. Okay, so what are ribose switches? Okay, ribose switches are actually RNA molecules. It is RNA that have the capacity to regulate gene expression in the complete absence of additional proteins. Okay, so these RNA switches include thermosensor. It can be regulatory antisense short RNA, and it can be ribose switches. Okay, so what are ribose switches? They are highly conserved. Okay, highly conserved maknanya the sequence are similar within, for example, different plants. Okay, highly conserved structures. Typically, okay, the location of ribose switches is actually terletak within the untranslated region of mRNA molecules. Kejap, hari ni SPM keluar kan? Itulah, kawan saya ada ni, anak dia dapat semua A kecuali satu B plus. Dia baru message Liza anak dia. Alright, okay. Alright. Okay, so So the ribose switches, the location of ribose switches is actually terletak dekat within, you tahu dekat mRNA kan ada untranslated region. Okay, so ribose switches are actually terletak kat this area. Okay, so they regulate gene expression of the adjacent open reading frame by direct association with small molecules. Okay, without participation of protein factors. Okay, saya tunjuk gambar aja Okay, so before that, you kena tahu characteristics of ribose switches, how they regulate gene expression. Okay, so first ribose switches regulate their cognate open reading frame expression without participation of any other molecules. Okay, so this character is very beneficial because it will allow ribose switches to act immediately to changes in intracellular metabolite concentration. Okay, and ribose switches, another characteristic, they can upregulate and they can also downregulate gene expression. Okay, so which enables the cells to activate or repress metabolic pathway while keeping adequate levels of metabolite. Okay, so for example, nanti kita akan tengok how ribose switches regulate production of thymine, okay, which is vitamin B1 in plant. Okay. All right, so these are the mechanism. Okay, so all right, so you imagine kan dekat untranslated region of mRNA. Okay, so you ada mRNA kat sini. Okay, for example, so for ribose switches, there are two different domains, two important domains within a ribose switch. Okay, the first domain we call as aptamer. Okay, so this, you imagine this is mRNA. Okay, so this untranslated region of the mRNA. Okay, so you bayangkan dekat ribose switch tu dia ada the first domain what we call as aptamer. Okay, domain, and then another part we call as expression platform. This aptamer domain is where the ligand, okay, akan bind. Okay, this is there to this minus MM, which is the metabolite lah. Okay, so M ni represent the ligand lah. Okay, so the aptamer part is where the ligand akan bind. Okay, while the expression platform is capable to change to the structure of terminator or anti-terminator structure. Okay, so you nampak without this kalau tak ada ligand yang akan bind to the aptamer, okay, so this is the structure of the ribose switch. Okay, alright, but when the metabolite or the ligand bind to the aptamer, nampak kan M ni, okay, dia akan change the structure of this expression platform. Okay, so nampak Okay, so this akan nampak. Okay, so this diagram, okay, akan tunjuk lagi better lah. Okay, so you have this apa tadi, aptamer domain, okay, and also the expression platform, okay, similar. Okay, so in the absence of the ligand, okay, so this is absence of ligand, tak ada ligand bind to the aptamer. Okay, so RNA polymerase transcribe the unbound aptamer domain, and in the default conformation, okay, in the default conformation, the expression domains adopts what we call as anti-terminator conformation. So this is anti-terminator conformation. Okay, so the transcription continues through the expression platform and into the coding region. So the mRNA will be transcribed. Okay, right. But in the presence of ligand, okay, or metabolite tadi, so for example, the ligand, it will bind to the aptamer, you know what's sini. Okay, so when the ligand bind to the aptamer, it will induce alternative conformation in the aptamer. Okay, so the platform, the expression platform, okay, it will induce a change in the expression, creating the terminator conformation. Okay, so RNA polymerase, okay, is effectively blocked, and it will the transcription will stop. Okay, so this is the mechanism of ribose switches. One example. Okay, any question? Tak ada?
No, Doktor.
Okay, clear? Alright. Okay, right. Okay, you pay attention Saya ada question dalam topik saya hari ni, pagi ni. Alright, so I will give you an example which is thymine ribose switch. Okay, so the thymine is TPP ribose switch. Okay, thymine ferrophosphate, okay, ribose switch, or also known as the Thai box, is actually the most abundant ribose switch found in living organism. Okay, so the a TPP ribose switch is actually the only ribose switch can be found in prokaryotes, archaea, and also eukaryotes. Okay, so in all cases, the binding of TPP to the RNA molecules downregulate the expression of thymine biosynthetic gene and thereby decreases the TPP biosynthesis. So bila ada ligand, TPP ni the ligand, or TPP ni bind to the ribose switch, okay, it will downregulate the expression of thymine biosynthetic genes and it will decrease the TPP biosynthesis. Okay, although the sequence of the TPP binding domain is highly conserved between prokaryotes, archaea, and eukaryotes, the gene regulation mechanism of TPP ribose switch varies between them. Okay, how they regulate the mechanism is actually different. Okay, saya akan bagi table nanti. Okay, so this reflect high capacity of this element to develop new gene regulatory processes. Okay, and the TPP ribose switch is the only ribose switch that has been identified in eukaryotic organism. Okay, all right. Okay, so because kita study plant, so I will give you contoh ni lah TPP. Okay, so this ribose switch ni is actually close to my heart because my labmate masa PhD saya memang buat ribose switch on the TPP. Okay, and also this paper is actually from her. So she's now a lecturer in UPM lah. Okay, so I said to her that I'm giving example based on your work lah. Okay, so this is her work lah. Okay, so tadi kita cakap TPP kan. Alright, so this is how the involvement of a TPP, okay, in apa ni, synthesis of thymine. Thymine ni is adalah vitamin B1. Okay, so kita akan tunjuk you. Okay, so kita tadi cakap, alright, the sequence of TPP binding domain is highly conserved between prokaryotes, archaea, and eukaryotes. But the mechanism is totally different. Okay, so kita tengok contoh Okay, so between bacteria also, there is also difference. Okay, for example, the location of the ribose switch is actually at 5' UTR, semua ni 5 UTR between gram-positive bacteria and gram-negative bacteria. Okay, so the mechanism, okay, how it regulates the expression is, for example, in the condition of low TPP concentration, okay, so in low TPP concentration, it form an anti-terminator ni lah structure, anti-terminator hairpin. Okay, right? And with high TPP, okay, so bila TPP bind to the aptamer or the binding domain, okay, it will form terminator. Okay, and this will lead to premature termination of transcription. Okay, so this is in gram-positive bacteria. Okay, while in gram-negative bacteria, totally different. Okay, the location of ribose is similar to 5' UTR. Okay, but in low concentration of TPP, okay, it form anti-sequester releasing the Shine-Dagano. Okay, dia akan Shine-Dagano anti-sequester. Dia akan form Shine-Dagano anti-sequester. Okay, and then the translation is being initiated. Okay, but while when TPP is in high concentration, okay, it will bind to the binding site. Okay, the formation sekarang akan jadi kepada shine diagonal sequester, okay, which lead to the translation initiation repression. It will repress the translation. Okay, so in fungi, different mechanism. Okay, so kita tengok in plants. Okay, the TPP, okay, the ribose switch TPP is located in 3' UTR, bukan 5' UTR. Okay, different. Okay, right? So in low TPP concentration, okay, dia bukan form pula macam anti-terminator. There is what we call as retention of intron. Okay, kena retention of intron and presence of poly-A tail signal in the mRNA. Okay, so it will lead to the formation of short and stable mRNA transcript. Okay, but in high TPP, okay, TPP akan bind to the aptamer or binding domain. Okay, it will cause the intron to be spliced. Okay, and generation of unstable mRNA transcript. So bila this mRNA transcript tak stable, okay, transcription will cease. Okay, so this is the different mechanism of regulation by ribose switches between the kingdom. Okay, right? So I'm just giving an example. Okay, so in plant kingdom, okay, based on the plant sequence database, okay, scientists have identified TPP ribose switch in the 3' UTR lah, macam kita cakap tadi is different dengan archaea and also in animal, where the ribose switch is being found in 5' UTR, but in plant in 3' UTR of the thymine C synthase genes. Okay, so the TPP ribose switch is being found in Thai-C genes. Okay, so Thai-C ni, okay, involved in the production of thymine. Okay, kita just gambar ajar ni nak tunjuklah kat mana function of Thai-C. Okay, so Thai-C is actually involved in the pyrimidine branch. Okay, for the production of thymine is involved two branch. One is pyrimidine branch and thiazole branch. Okay, so to produce thymine. All right, thymine pyrophosphate TPP. So this is TPP. Okay, so they found the TPP ribose switch in the 3' UTR of thymine C genes. Okay, so remarkably, not only the TPP ribose switch itself, but also the entire 3' UTR of this gene is actually conserved. Okay, so in all cases, the stop codon is immediately followed by a constitutively spliced intron, and the following exon includes a polyadenylation signal and a potential splice site. Okay, tadi kita dah nampak kan? Okay, ada intron splicing or intron retention, and also ada poly-A tail. Okay, and the TPP ribose switch is located actually 70 base pair downstream of the polyadenylation signal, and the TPP is actually 150 base pair long. Okay, and the last exon of this 3' UTR is a variable leg and can extend over 1 kb. Okay, so the two splice sites of the second introns are tightly linked to the TPP ribose switch. Okay, its 5' splice site can base complement the P4, P5 arm of the TPP ribose switch. Okay, and the 3' splice site is located within the TPP ribose switch P2 box. Okay, you tak ingat ni tak apa. Alright, so this is another gambar raja lah, okay, to show you the TPP ribose switch, okay, between eukaryotes. Okay, you don't have to remember. Okay, and also this is what happened in Thai-C 3' UTR lah. Okay, untuk tunjukkan apa yang saya cakap kat sini. Okay, right, so that's all untuk gene regulation. Any question? Ada question?
Tak ada.
No, Doktor.
Alright, okay. Alright, what I want to tell you, Dr. Latif sebenarnya baru bagi saya a few minutes ago, result midterm U. Okay, so you nak tahu sebab kami kena keluarkan CAM by 19.